Thank you, Anna, very much for that introduction, also for the invitation to come here. It's a great pleasure, a privilege to be able to address you at this annual conference. The world is full of species, of animals, of plants, and of microbes. And for us evolutionary biologists, one of the most fundamental questions, in fact, two of the fundamental questions confronting us, is why are there so many species? And why are they so different from each other? Now, these questions have attracted a lot of attention in the last, I don't know how many decades, 4 or 5 anyway, from systematists, from geneticists, from paleontologists, from a lot of other disciplines within biology, in order to answer these extremely broad-ranging questions. Those of us who ask those questions find ourselves in the footsteps of two Victorian British 19th century naturalists, Alfred Russel Wallace and Charles Darwin, because they gave us a theory of evolution by natural selection. They also gave us a theory of species multiplication. And in addition to those, they gave us indications of how we could test the implications in natural environments. So here are a couple of quotations to give you a flavor of how they gave us these pointers. From Russel Wallace, first of all, Alfred Russel Wallace. He wrote, "I should like to take some one family to study, thoroughly, excuse me, principally with a view to the theory of the origin of species. By that means, I'm strongly of the opinion that some definite results might be arrived at." Darwin said more or less the same thing at greater length and in more detail as follows. In his writing of a little bit later, 1859, he wrote, "Those forms which possess in some considerable degree the characters of species, but which are so closely similar to some other forms, or are so closely linked to them by intermediate gradations, that naturalists do not like to rank them as distinct species, are, in several respects, the most important to us." Well, to give you a flavor of the kinds of organisms that can reveal to us the reasons for evolution, we could pick a whole variety of creatures and organisms ranging from viruses to grasses to whales. But what I'm going to do is to introduce two groups of vertebrates. First of all, anolis lizards in the Caribbean archipelago, 150 species in the same genus, all derived from one or a very small number of common ancestors. And my next example is cichlid fish from the Great Lakes of Africa and in the neotropics. In this case, it's literally thousands of species, all derived from a very small number of ancestors in a relatively short space of time. Now, our chosen group is much smaller, less dramatic. Darwin's finches, approximately 14, maybe a few more species, but a very small number, that have originated in the Galapagos and diversified in the Galapagos Archipelago in the last couple of million years, according to mitochondrial DNA dating. So, in other words, relatively recently. And the question they pose to us is how and why? How do we get so many species so rapidly? And key to the answer is the question of how species evolve. That is, how do we have two species derived from a common ancestor? The process here is depicted as a branching process which results in the inability, eventually, of the derived populations to interbreed even if they attempt to do so. Darwin proposed that spatial separation, geographical separation, would favor the initiation of this process. And what I'm going to do is to outline his ideas with modern language using the Galapagos as the template for the ideas. So they involve three things. The first is dispersal. The second is evolutionary change in separate locations. And the third one is the coming together of previously separated populations and further evolutionary change, divergence from each other. The responsible factors are pervasive, random genetic drift on the one hand, and natural selection on the other. And why? Because these islands are not identical in the kinds of challenges they present to organisms that occupy them. And in the case of the coming together of two populations in sympatry, there's a further divergence under natural selection, resulting in the minimization of two things. One is interbreeding, on the assumption that the products of interbreeding are relatively unfit. And the second one is a minimization of competition for food. Character displacement is what that is referred to now, ecological character displacement. And I will come back to this with an example towards the end from our study on this little island of Daphne, right in the middle of the archipelago. Now, I don't want you to think that the evolutionary drama that is played out on the stage of Galapagos is in a fixed theatre. On the contrary, the Galapagos Archipelago is one of the most dynamic environments on Earth. They are volcanic in origin, formed, most of them, by a hotspot beneath the island, western island of Fernandina, by puncturing a plate that is moving south-southeast across the archipelago in this direction at the rate of 60 million years, 60 kilometers per million years. And as they move away from the hotspot, they subside until eventually they're below the water altogether. So that is a dynamism that is taking place over a long period of time on a long temporal scale. In addition, the sea level fluctuates according to whether, over the last million years anyway, according to whether water is locked up in ice at glacial maxima or is released as water in interglacials. So relative to the water level, which is going up and down, the islands seem to be going up, higher or lower, spreading outwards, laterally or compressed, spreading outwards even to the point of coalescence. And I'm going to illustrate that dynamic with three pictures. The three pictures are bathymetry, the array of islands nowadays, and then two glacial maxima when a large amount of water was locked up as ice in polar ice caps. First, the previous maximum, and then the greatest maximum in the last million years. So it goes as follows, there and there. And then in reverse, you can just imagine the water level rising as the ice melts. We go backwards to where we are at the moment. So both the evolutionary play and the theatre, the environmental theatre itself is undergoing a change from time to time on this scale. So about 40 years ago, we began a study on this little island of Daphne Major in the attempt to investigate locally the processes that must have occurred globally across the archipelago to give rise to all of the species that we now know today. The island's only about 3/4 of a kilometre long and about 120 metres high. It's never been settled by humans. The environment is pretty sparsely vegetated, but it is at least in a pristine state. There are 4 species on this island in the groundfinch genus Geospiza. There won't be very many scientific names for you to remember, but there will be 2 or 3 or 4. There's a small and a medium and a large groundfinch, and then bottom right, a cactus finch with a relatively pointed beak. This is going to become more obvious in the next picture here. A large, a medium, and a small groundfinch, a cactus finch with a pointed beak, and even more clearly, perhaps, in addition to this, in this plot where we have beak width plotted against beak length for the average population of all of the populations, rather, sorry, pardon, here. So each one of these dots is an average of a population, and there's several populations of Fuliginosa. Now I'm going to switch to the Latin name. The small, medium, and large ground finches are Fuliginosa, Fortis, and Magna-rostris. And here is Scandens, the cactus finch, off at one side of that line of allometry with relatively pointed beaks. Fortis and Scandens are the most common species on the island. Fortis is this one down here, has a population size of about 100 breeding pairs. The Scandens is this one here, and that has a breeding population of about 50 breeding pairs. So a little bit about the natural history of these birds, first of all. Fortis is a sparrow-like bird, a generalist. In its feeding, it feeds a lot on seeds, and those seeds come from a variety of plants. So to that degree, it's a generalist, but it's also a generalist to another degree as well. By banding a lot of birds, capturing the birds, banding them with individual combinations of bands, and then measuring them and releasing them, we were able to observe and determine that birds seen feeding only on small seeds were relatively small in big size, definitely so in comparison with those feeding on small, medium, and large seeds. So the species is a generalist, but to some degree there's an amount of specialization according to beak size and food size. The other common species is Scandens, and this differs in that the species and all individuals are specialists on cactus. They do feed on seeds as well, but they all do the same thing. So they all probe into a puntia prickly pear cactus flowers and extract nectar and pollen as food, but they do something else as well. When the fruits are formed, they, with their pointed beak, they hammer holes into the side of the fruit and extract the seeds and consume them and keep going until they more or less exhausted the fruit. So that's a specialist species. All right, now to understand evolution, we need to be able to understand how genotypic variation is converted into variation in fitness via the phenotype under the influence of the environment. And I'm going to take these first two relationships here, this one and this one, separately, one at a time, and start with this one and ask the question, how, knowing observable phenotypic variation, can we find out about unobserved cryptic underlying genotypic variation that is responsible in part for that phenotypic variation? Well, we addressed this question very many years ago by using the techniques of quantitative genetics, which involves comparing parents and offspring in their measurements on the assumption that at least part of the resemblance of offspring to their parents is due to the transmission of genes from one generation to the next. So we did that with a very large number of families of birds, and we found a very strong relationship between, in beak depth in this case, Fortis above and Scandens below, between the average of the offspring and the average of the parents. The line, the slope, is a statistical measure of the degree of relationship. It is an estimate of the heritability or degree of heritable variation in this trait. And it's high. The slope is between 0.7 and 0.8 for these two species. And to give you a little bit of context there, that's about the same degree of heritable variation as we have in our population in height. Human height has a heritability in that range. So we wanted to understand the genetic basis of variation in more detail, and to do that we collaborated with developmental geneticists. We are not developmental geneticists, so we have collaborated with Cliff Tabin and Arka Abshanov at Harvard University, who are the experts, the specialists. providing them with embryonic material to probe the development of various stages of development of finches using the candidate gene approach. And they discovered, initially, their first discovery was one signaling molecule, BMP4, that varies markedly between Magna rostris, the large groundfinch, and the one with the shallowest beaks here, scandens. It affects beak depth expression, the depth, meaning in this plane, of these species, and the results are shown graphically here with the staining indicating the expression of BMP4, bone morphogenetic protein 4. And what I'd like you to do is to interpolate between the deepest and the shallowest. There are 4 other species in between, and they have an exactly graded expression profile relative to their phenotypic profile. To make sure that this was not just a correlation, but there was a causal relationship between this signaling molecule and the phenotypic trait, beak depth, they did transgenic chicken experiments, putting a Magna rostris gene in chickens and finding, in fact, that they could get expressed a Magna rostris-like beak in a chick. Well, this is a large body of work. This is the beginning, and I'm going to summarize give you a summary of the total contribution of this research effort. And the bottom line is this, that each of the 3 beak dimensions, depth and width and length, can be altered by the alteration of expression of different signaling molecules. And it goes like this. The bone morphogenetic protein number 4 expresses or regulates the covariation of depth and width. Length is independent of those 2, regulated by another signaling molecule, calmodulin, in a different signaling pathway, independent of depth and width. And what about width, independent of depth and length? Well, that is regulated by a different pathway, I've shown up at the top here, a little bit later in development when bone is being condensed. So, once again, it is relatively easy to alter any one of these dimensions independent of the other by an appropriate genetic alteration of the expression of these signaling molecules. So back to this diagram, what about now the way in which variation in fitness maps onto phenotypic variation? And I'm going to start by emphasizing that for much of the time there is no variation in fitness in relation to variation in phenotype. And here is a single example to illustrate the point. Beak depth is the trait. The frequency for the species, the medium groundfinch, from 1989 to 1990, fitness was a straight line function of beak depth, which means to say that birds of small, medium and large beaks had equal fitness over this period of time. And indeed, the specialist species, scandens, shows no variation in fitness, average fitness, across the whole of the 40-year period in this particular trait, beak depth. And I think for many people this is not at all surprising. We don't see evolution taking place, so I think our standard view of the world is that it is in evolutionary terms static. So, for example, the rabbits and the dandelions that we see outside don't look any different from the rabbits and the dandelions that we remember in our childhood days. The situation with forties is completely different. There are, 40 years, 3 periods of major perturbation under selection, and I'm going to deal with each one separately because the factors involved differ. The direction of selection and the reasons for selection varied across this period. But there are common denominators. The common denominator is a drought. On each occasion, there was a very severe drought, resulting in starvation and mass mortality of finches. Let's start with the first one, the 1977 one, in which there was almost no rain throughout the whole year. 4 out of 5 fortis died during this period, and during this period, average beak depth increased systematically until the rains resumed in 1978, and there was a further production by plants. And the reason for that differential mortality, not emigration but mortality, was that initially the large abundance of small, easy to handle, easy to crack open seeds was depleted to the point where finches had to turn increasingly to the large and hard seeds indicated by that little diagram picture up at the top left of tribulus, a fruit, woody fruit that is difficult to crack open. Well, only the birds with large, deep beaks were able to crack open those fruits and get at the 5 seeds on the inside. They survived. The birds with smaller beaks did not. So natural selection occurred from late 1976 through to the beginning of 1978. Not only that, but given the fact that the trait is heritable, there's genetic variation underlying the phenotypic variation, the degree of evolutionary change from one generation to the next, from the selected generation to the offspring generation, could be predicted very closely by a combination, the product of the heritability of the trait and the strength of selection, the so-called simple breeders equation. Can be, as I said, can be predicted, and that's indicated by that star up there, which is the position of the average offspring produced by the survivors. The difference between the predicted value and the observed value was very close. So in summary, in this event, natural selection occurred on a heritably varying trait for mechanical reasons that we could understand when the environment changed. The next event involved something very different. A complete change in the vegetation as a result of an extraordinarily strong El Nino event of 1983. El Nino is a phenomenon of oscillation of both pressure and temperature, sea surface temperature across the Pacific Ocean. And in this diagram, the intensity of sea surface temperature is indicated in red. The opposite of very low temperature, nicknamed La Nina, is shown on the left in blue. And you can see from this pattern that there's this oscillation back and forth between these extreme conditions with a periodicity of about 4 to 7 years. Well, the 1983 El Nino, according to coral core data, was the most intense El Nino in 400 years. And it certainly affected the Daphne rainfall pattern with that enormous amount of rain, with consequential effects on the plants. And I'm now going to illustrate, but before I switch to the pictures of plants, I'd like you to notice that 2 years later there was a complete drought. So we'll start with a picture of the so-called, we euphemistically call this the plateau, on the island. It's the dry season. You see cactus bushes in green and leafless trees at the back and almost nothing in the foreground. The rains come and the island turns green, and in the foreground little plants start to grow up, and amongst these plants is a tribulus, the plant whose fruits made the difference between survival and non-survival of birds with different beak sizes in 1977. And at the bottom you could see an exploited fruit there. Finches have torn off the wood. Right, so the rain came down and it continued for not just 1 or 2 months, but 8 months. It converted the island into a completely different vegetation composition from the preceding one, with small seed plants having an advantage over the large seed plants. And in particular, tribulus got smothered by this rampant growth of, first of all, grasses, and then vines growing up over the bushes, over the trees. And even after, 1 or 2 years later, we could see the effect of this transformation of the vegetation in the form of vines draped over the trees. So the island became converted from basically a large seed producer, to simplify a little bit, to a small seed producer. And under these altered conditions, now the selection switched to the opposite direction. And instead of, as in 1977, individuals with large size having an advantage, as shown by these bars here, which represent selection coefficients on wing weight, wing, tarsus length, beak length, depth and width. Now in the mid-80s, the selection coefficients were in the opposite direction, and I'll interpret by telling you that that is virtually no selection in contrast to selection downwards on depth and width. So all traits went down, or almost stayed constant in the case of beak length, but because there was a difference in proportions of the beaks, the beak shape underwent a change, so that birds with small beaks but with more pointed beaks survived better than those with the average condition before the drought arrived. So the third selection event again involved a very different factor. It was the arrival of another species on the island, Magnorostris, the large ground finch. You can see from these pictures what a wonderful tool the beak is for cracking open the large and hard tribulus fruit. I mean, it just looks so much more powerful than as the tool possessed by the large members of the medium ground finch, Fortis, and the small members. So what happened as the drought, the next severe drought of 2004 elapsed, what happened was that there was a very large amount of mortality amongst the medium ground finches, Fortis on the left, and then it became critical which birds could crack open the tribulus seeds. Now, Magnorostris is much more efficient than the large members of the Fortis population, and so these died much faster than Magnorostris. Also, the small members of Fortis died, but they died also at a slower rate. These are the ones that were selectively penalized. The net result then was the average beak size went down. It went down drastically, and it remained down for a number of years. And in Rosemary's talk, it will become more evident why it remained down rather than bounce back up to the preceding state. The final point that I want to make about this event, it is an example of character displacement, a divergence of Fortis and Magnorostris as a result of competition. Fortis, on average, diverged away from this superior competitor, Magnorostris. Final point that I'd like to make about this is that just recently we have uncovered a gene with major effect on this key trait of beak size involved in character displacement. And we've done this by collaborating with another group of experts, and they are, sorry, that was that, right. Okay, so they are a group in Sweden headed by Professor Leif Andersson here at the University of Uppsala. They have all the techniques to do what we wanted to see them do, which is to, first of all, sequence the genomes of all of the species of Darwin's finches, and then set up a comparison to look for genes that underlay variation in beak size. So we first of all started with size, overall size, as a factor of interest. Now, they compared, if you can see up at the top here, large with small birds, large with medium birds, and medium with small birds. All possible ways of comparing species of large, medium, or small size in pairs. And they discovered that there was one of the several differences that stand out here, but one in particular was a real standout. It is this locus here, with the prominent element being HMGA2 as the correlate of size. Now, HMGA2 is a transcription facilitating factor. It's not a transcription factor itself. It's not involved in transcription directly, but it facilitates transcription in humans. And it has influences on such things as overall size, but also craniofacial features, including dental features and intercranial distances. So there's a very good presumptive case that it has an effect upon overall size in the finches as well as the beak size. Well, it turns out that this 525 kilobase region varies in a plus or, not a plus or minus, a 1 or 2 discrete alternative state in the finches. So if you look on the left-hand side, you'll see HMGA2 at the top here. Within this region, HMGA2 is on the left. All of the large species are shown in red at the top. All of the smaller species are shown in blue at the bottom. And the intermediate ones have both types. So there are two haplotypes, a large and a small one. And those haplotypes are segregating in the Fortis population. And not only that, their association with beak size is exactly the same as the overall association with body size in the interspecific comparison. So there is the indication that this is so. A very strong relationship. Look at the p-value up here. With the what we call, on the basis of the interspecific comparison, the L genotype in homozygous form being associated with large size, and the SS homozygous being associated with small size, and the intermediate, as expected from additive inheritance, be intermediate in average size. So the crucial point here is that that genotype underwent a very strong frequency difference in the course of that character displacement event of 2004. And here are the data that show that, with the survival of the LL genotypes being approximately 1/2 of the survival of the SS genotype. That is, the small birds survive better than the large ones, and the genotype matched the phenotypic change that took place as a result of selection. And in allelic terms, the S allele shifted from 37% to 62% in this one single selective event. So a major genetic influence on a polygenic trait resulted in a major genotypic shift in the composition of the population, which is remarkable and unexpected. So I'm going to summarize at this point with 3 conclusions about the role of natural selection. The first is that selection occurs when the environment changes. Usually it doesn't happen, but at least detectively. The second conclusion is that the evolutionary response to selection from one generation to the next is predictable, and I'll illustrate that with the data that show this, with the predicted values being plotted on the vertical axis against the observed ones. The contrast is strongest between these 2 events that I started and finished with, but also present in the other years as well. And the third conclusion is that long-term I'm now returning to the theme of speciation that I started with. Long-term studies are needed in order to be able to understand the process of speciation in contemporary time. And I'm illustrating this with a diagram that plots for Fortis two beat dimensions against each other. Each point is an annual average. The population started in morphospace here and finished there. So you can think of this as a speciation trajectory from, if I can hold the line, anyway, from the center down to the bottom left. Now, I'm not claiming that that is a new species here. What I am saying is that that direction of change would lead to a species out here if it continued very much longer. But in reality, that's not the way evolution takes a species from one state to another. The data show that it's a very erratic pathway according to the way in which the environment changes from one time to another. So we had the population being propelled upwards along a line of allometry, interannual line of allometry early in the study, and then another allometric shift in the middle of the study. It was a relatively small one, but highly significant change in beak shape, to a position on a parallel line of allometry where the populations have wandered about a little bit stochastically from year to year, going nowhere directionally, until the next major drought drove the population down into this region there, where it has stayed ever since. As I say, on the way towards becoming a new species, but staying in its position as a result of that erratic oscillation of selective forces. Now I'm going to hand over to Rosemary at what is the easiest point to get on the island.
So I'm going to begin where Peter left off with this diagram. Each of these spots was 1 year, and the time when there was a shift from one allometry to the next line of allometry was the year 1986. What actually went on in this year to cause this enormous shift in bill shape? So you can often quite easily get a shift in bill size, but to get a shift in shape is altogether more difficult. And the answer is introgression. There was gene flow from Scandens to Fortis, which began at this time. And so I'm going to deal with the causes and evolutionary consequences of introgression or genetic exchange between species. And then what I'm going to tell you about is another very exciting part of our study, which is a formation of a new lineage, which we followed from its inception all the way through for 6 generations. First of all, again, I'm going back to one more of Peter's slides. And this is the Y diagram where Peter explained that a population can split into two, go onto 2 different islands with different ecological conditions, and then they can evolve along that way through the process of natural selection and evolutionary responses to natural selection until they get to the point when they cannot interbreed, even if they came together on secondary contact for reasons of genetic incompatibility. Now, at this point, from when their lineages are diagnosably different to the point when they cannot breed for reasons of genetic incompatibility, Alan Wilson has shown can take in birds on average 32 million years. The same is in fish and the same as in reptiles. It's an order of magnitude less in mammals. But Darwin's finches, as Peter has told you, is only, the whole radiation is only about 2 million years old. So we would expect, instead of genetic incompatibility between the species, we should expect that the species are being kept apart by pre-mating barriers. So what are the pre-mating barriers? Well, all Darwin's ground finches are similar in plumage. Males are black, females are brown. They all build similar nests. They all, as far as we can tell, have similar courtship displays. But they differ, as Peter has said, in morphology, particularly in size and in beak size and shape, and they also differ in song. So the first thing we did when we went onto the island was to ask, we can discriminate between the species on morphology and song, but can the birds do this? So we took museum specimens, stuffed museum specimens, took female fortis, a female scandens, put it on either side of a pole, took it into a territory and asked the territory owner, can you discriminate between a female of your own species and a female of another species? And the answer was a resounding yes. They vigorously courted the female of their own species, even though she was just a stuffed museum specimen. And they completely ignored the female of the other species. So again, we asked, can they tell by song alone? And so we recorded song in the absence of any morphological cues. We played back song, we played back fortis song. Fortis came into the loudspeaker, scandens completely ignored it. We played back scandens song, scandens came into the loudspeaker, fortis ignored it. So clearly they can discriminate between their own and their another species on the same characteristics that we use. Song is very different between fortis and scandens. Fortis is a modulated song, scandens a song of repeated notes, and there's variation between individuals, but always on these species, always on the same species variation, similar variation. So we also know something else about song, and that is from work by Bowman in the 1950s, where he was able to take captive birds over to actually San Francisco, and he had a whole breeding population of many, many species of Darwin's finches, and he found that in all of them, song is learnt from an adult male in association with appearance during a very short receptive period early in life between day 10 and day 40. Now, this corresponds to the last few days when the birds are in the nest, to when the birds are out of the nest being fed by their parents, and all this time the father, the male, is singing. So it's not too surprising that they learn almost perfect renderings of their father's song. Now, when they come to, well, I should say, once they've learnt this song, it is retained for life. Life can be as long as 17 years. And when they're adult, they mate according to the species song learnt early in life. So this, of course, constitutes a pre-mating barrier to interbreeding. But we can ask, this barrier is based on learning song in association with morphology. So does it ever leak? And of course, the barrier is breached if the young bird hears and learns a song of another species during this short sensitive period. And this does happen. Scandens is a little bit larger than fortis. It's more aggressive. It frequently takes over, well, not frequently, it can take over the fortis nest. Normally it turfs out all the eggs. Occasionally an egg is left behind. The egg grows up as a fortis in a scandens nest and learns its foster father's song. Also happens if the male dies and the female is left to rear the offspring. And if the natal neighbor is another species, then it will learn that species song. So we get occasionally birds learning and singing when they grow up the other species song. Now, the colonization of Daphne, which by Magnerostris, which Peter mentioned, gave us an opportunity to test this. Magnerostris is fortis sister species. It's the closest relative, but it's much larger. It's almost twice the size at about 32 to 34 grams than fortis, 17-gram bird. It's also much larger than scandens at 22 grams. It's extremely aggressive and it has a loud penetrating song. But over the years, we had 8 fortis and 2 scandens that learnt and sang a Magnerostris song. So we have the Magnerostris song down here, typical Magnerostris song. Here's one of the fortis that sang a Magnerostris song up here. This is the normal Magnerostris song. The scandens singing a Magnerostris song, a normal scandens song. And none of them actually bred with the Magnerostris, so why not? And because the song, as you can see, is pretty close to a Magnerostris song. In fact, it's so close that when these little birds opened their mouths and sang a Magnerostris song, a Magnerostris would just whip in as if from nowhere and just beat them up. So these poor little birds never survived to breed. So it seems that size matters. When the size difference between the species is very large, the actual barrier is robust. Even learning a Magna Rostra song didn't lead to interbreeding. But when the size difference between the species is small, as it is between scandens at about 22 grams and Fortis between 17 and 18 grams, learning another species song can lead to hybridization. And this happens about 1 to 2% of every breeding session. So we had a series of hybrids between Fortis and scandens, and we could ask then, Fortis and scandens are not sister species. They're in the same genera, but they are not the closest relative to each other. So we can ask, how fit are the hybrids? Are they viable or are they fertile? Is there any sign at all of any genetic incompatibility? Well, at first we thought there could be, because in the first 10 years of our study, we had a series of these hybrids, and none of them survived the dry season to breed. But then we thought, in the dry season, the predominant seeds were these large, hard tribulus seeds that Peter talked about, and also large, hard cactus seeds, puntia seeds. We watched the hybrids try to crack them. They were unable to crack them, and they could crack the Apuntia seeds, but took about 3 times longer, 2 to 3 times longer than scandens to do so. So it could be that the intermediate beak size was inappropriate for the amount of food that was available. And this seems to be the case, because after the island was converted to an island with a massive production of small soft seeds, then these hybrids began to survive. But we can still ask, how well did they survive relative to the parental species? And so this is after, well, in 1983, when there was a large amount of small soft seeds available, we had a production of over 1,000 Fortis and over 500 scandens and 12 hybrids. And again, in other years, when there was a large production of small soft seeds, how well did they survive? So these lines represent the parental species, Fortis and scandens. If there had been any incompatibility, the survival, we thought, line of the hybrids should lie below the line of the parental species. But instead, what happened is that the hybrids survived as well as, if not better, than the parental species. They produced as many eggs when they bred, as many eggs, as many offspring, and as many recruits. So we had no evidence at all of any genetic incompatibility. They did not breed without each other. Instead, they backcrossed according to their song type. So you can imagine Fortis here had learned as a young bird a scanden song. He would mate with the scandens and produce a hybrid which also sang the father's song, mated with the scandens, and produced the second backcross generation, mated with the scandens, and so on. And the same down the Fortis side. So we got, through this process, a trickle of genes going from one species into another. And it went both ways. So what happened is that they began to converge on each other. So these plots are beak depth over beak length. For the first 10 years of our study, each dot within these circles is a bird. And so for the first 10 years of our study, Fortis was clearly different from scandens. And the two species introgression continued, 87, 1991, and up to 2012, and convergence in morphology continued. Another way of looking at this is looking at the difference between the beak size difference between the birds, between Fortis and scandens population. This is a point when introgression began, and you can see here that Fortis and scandens are becoming much more similar in their beak difference. And if we were to extrapolate from there, in 2057, when we will be long gone, but many of you that are here will be here, then they should be identical. if nothing happens. But of course, this is not typical of the Galapagos. Something could easily happen. But of course, they also converge genetically. Now, this plot was made out of our microsatellite loci, which showed that they were converging genetically. But we then, and at this point, just to say that this was in 1986 when the convergence began, then from there onwards, Fortis' bill shape became more pointed, because introgression was continuing for the next 30 years, and the beak shape became more pointed. So we can ask, and this is, again, this point back to this slide. So we can now ask, do we know anything about the reason for this? Do we have any genetic underpinning of the reason for the beak change in Fortis? Why were they becoming more pointed? And here we go back again to our collaboration with Leif Andersson and his group in Uppsala, because they found a couple of years ago that there's this gene ALX1, which encodes a transcription factor. This gene is very important. It's highly conserved. In fact, it is even in humans, in the cranial-facial development of humans, and a mutation in this gene causes cleft palate in humans. In the birds, in the finches, it comes in two haplotypes. The ALX1 blunt haplotypes is associated with blunt beaks, and the ALX1 pointed haplotype or pointed variant is associated with pointed beaks. Now, they got to this by sequencing, through the sequencing of the genome of all the species, and by, again, comparing species with pointed beaks and those with blunt beaks. And there are very few with really blunt beaks, Magnorostris and Conirostris on Espanola, and they were homozygous for the blunt haplotype. And then most of the other birds have these pointed beaks, and they have the ALX1 pointed haplotype. But fortunately, Fortis on Daphne actually had both. So we then sent a large number of blood samples over to Uppsala, blind, and said, to find out if the same haplotypes were segregating in Fortis, just as they were segregating between species. So we sent 62 blood samples over to Uppsala, and they sent back the results, and it fitted beautifully. So the homozygous blunt haplotypes here were associated with the blunt beaks, homozygous pointed haplotypes were associated with the more pointed beaks, and the heterozygotes were intermediate. So we then asked the obvious question, did the ALX1 pointed variant, was that introduced from scandens into Fortis, and did it contribute to the Fortis more pointed beaks? So again, what we did is we went into our -80 freezer, and from this time we had 8 hybrids and 79 Fortis non-hybrids. And just to remind you that Fortis had both these haplotypes in the population, but scandens only had, before introgression, only had the pointed haplotype, and also occasionally on the island, Fuliginosa, the small ground finches, comes over, that also only has a pointed haplotype. So these 8 hybrids were made up of 6 from Fortis scandens and 2 from Fortis fuliginosa. And we got back the result, and we found that the ALX1 pointed haplotype had a frequency of 0.75 in the hybrids and 0.44 in Fortis non-hybrids, which strongly suggests that introgression of the ALX1 pointed haplotype from scandens to Fortis most probably contributed to the Fortis more pointed beaks that were better adapted at exploiting small soft seeds. But of course, gene exchange goes both ways, as I mentioned earlier, and exactly what you would expect, the average beak shape of scandens becomes blunter over this time. And in both populations, they increase in morphological variation to a noticeable extent. All these photographs were taken in 2012. They're all scandens. The scandens at the top has this long pointed beak and looks exactly like and has the same similar measurements to the scandens before introgression. The other two scandens are much more fortis-like with a blunter beak, and there are many fortis like this after introgression. And many scandens like this after introgression. So we argued if such a bird flew to another island with different ecological conditions, their novel genetic compositions and phenotypic characteristics could be the starting point of a new evolutionary trajectory, leading to a new lineage. We'd argued this after we knew about introgression, and we never, ever expected to see it. But we did. What happened was, this is in the formation of the new lineage of finches, a bird, excuse me, a bird arrived on the island, and it was a very large bird. It looked like a scandens, looked like a fortis. Fortis is 17 grams. This bird was 28 grams. It arrived at a time when we had almost all birds, or we thought we had all birds on the island banded. So where did this bird come from? Was it a fortis? What was it? So we caught it, took a small drop of blood, and we matched up its genetic profile with all the birds on Daphne, and we found that it didn't match any of the birds on Daphne, and so we matched the profile, genetic profile up with all the birds, all the islands around Daphne, and found that it most likely, with a high probability, came from Santa Cruz, and it was possibly a Fortis scandens fortis backcross from Santa Cruz at a probability through an assignment test of about 90% probability. It also, to our delight, had a genetic marker. It was homozygous for this rare genetic allele at 183, and at one of our microsatellite loci. And when it began to sing, it had a completely unique song, never before been heard on Daphne, and actually never heard on Santa Cruz. So this is a puzzle. It took ages to breed, but when it did, it bred with a Fortis scandens fortis backcross born on Daphne. And then later it bred with two Fortis, and that was the only outbreeding that occurred. It produced a fair number of birds. All the birds had at least one copy of, excuse me, I'm going to, all the birds had at least one copy of the unique allele, and all the males sang 5110's song, which was passed down from father to son to grandson, etc. And we were at this situation when this 2.5-year drought arrived on the island, when almost 90% of the birds on the island died. All these birds died except for these two, the brother and the sister, which were highly inbred. So they survived, and when the rains came again, they bred with each other. They produced 26 offspring, all but 9 of them bred. So we had a daughter breeding, I'm sorry, a daughter breeding with her father, a son breeding with a mother, and the rest breeding with each other. This carried on, this inbreeding carried on for the next 3 generations. All these birds were homozygous for the rare allele due to the previous inbreeding. All males sang the 5110 song, and all were large. So from 5110, we had cultural transmission of song and genetic transmission. So is this new lineage behaving like a separate species? It's very much larger than its nearest relative, Fortis. There's a young bird at the top and Fortis down below. It differs in size, and in morphological space, it lies here between Magnarostrus and between Fortis. And this was a space that became expanded when the Magnarostrus outcompeted the large Fortis during the character displacement event that Peter told you about. They sing different song from all other species on Daphne. This is 3 generations of the big bird song: Fortis song, Scandin song, and Magnarostrus song. A very different song from all other birds on the island. The pairs hold contiguous territories, which they defend against each other. The blue dots are the territories before the drought, the red dots after the drought, and these territories overlap with Fortis, Scandins, and Magnarostrus territories, who they completely ignore and who are ignored by them. So in every respect, this population of big birds is functioning as a new species. Will it die out through inbreeding depression? Well, as you can see, it is surely inbred. So far, we see no sign of inbreeding depression. Will the genetic variation be augmented through genetic exchange? We think this is the most likely thing that might happen, but so far, and we've looked at this for this very carefully, so far we've not seen this happening. But whether it survives or not, this new lineage gives us insight into how a new species could arise and either persist or go extinct. So, given the origin of the new big bird lineage through interspecies gene flow, we then asked, is there any evidence of genetic exchange between species at earlier stages of the adaptive radiation? And I won't go into this in great detail. It's just to say that we used the Abba-Babba test or the D statistic to look for this after we had this new phylogenetic tree, which we had done from these whole genome sequences. And the answer was, I can go into this for anybody later, but the answer was yes, we did get, very much we did get signs of genetic exchange, which most probably contribute to the formation of new phenotypes and the development of new species in the past. So in summary, we started our work in 1973 with 2 various distinct species on the island, Fortis and Scandins. This is bill depth over bill length. And it remained this way for 10 years. Then we had this enormous El Nino event, the largest in 400 years, according to Coral Core data. Fortis and Scandins began to interbreed and exchange genes, and they began to converge. Magnarostrus, which arrived on the island at this time, built up a huge population. In the drought of 2004 to 2005, it outcompeted Fortis, as Peter told you, in its character displacement event. The big birds, which had been building up, just moved into this morphological space, and we've ended the situation here. So we've been very impressed how the coming together of 2 or more rare events in unusual combinations can trigger rapid and unpredictable evolutionary change. I mean, we had this enormous 1983 El Nino event. At the same time, we had the colonization of Magnarostrus. And during this enormous 2.5-year drought, which actually, again from Coral Cores, was the most severe drought in over 100 years, we got the survival of a brother and sister, 2 birds which just happened to be a brother and sister at a time when all the large Fortis had died. So our work has showed insight into 3 models of speciation. The Allopatric model, where we had the coming together of Fortis and Magnarostrus on secondary contact. We found no interbreeding, but a character displacement event which led to divergence. Then we have the underappreciated role of introgression in species formation. We had species by fusion, where we get Fortis and Scandins leading to one unique lineage of mixed genetic composition when Fortis and Scandins are remaining separately on other islands in the archipelago. And then we had a very exciting finding of speciation by integration in a new environment, which led to the development of a unique lineage, the big bird, what we call the big bird lineage. So I know that you all know this because this is your logo when I look on your webpage. And this very famous diagram of Darwin's from his notebook, where he was trying to puzzle out the relationship between species, and he wrote, I think. Would he now join up some of these lines and say, I now realize? Maybe. So we thank our very many graduates, postdoctoral fellows, our collaborators and daughters. None of this work could have been done without them. But all of us who have worked on the island would want to leave you with one message, I think, and that is a conservation message, that the environments and populations are dynamic, they're constantly changing and have been changing for a long time. So to sustain our environment, we must keep them both capable of further natural change. Thank you.
So I think I remember when I read the paper of the big bird lineage that the first female that bred with the immigrant was also a hybrid.
Yes.
Maybe that's why she could recognize the song, maybe the hybrid song as well. Was it that way or was it a different way?
But the song that this female came actually from, was singing a perfect Fortis song. I mean, came from a father that was singing a perfect Fortis song. So, and the songs, we don't get a mixture of songs. We never get a hybrid song. This hybrid song, we don't know where it came from, because we have given the number of people working in Darwin's finches on Santa Cruz. They can't find it in Santa Cruz. We can't find it in Santa Cruz. The song is a bit of a mystery. Yeah. We can speculate how it happens, but oh, I'm sorry. But actually, for people who didn't hear the question, it was about the hybrids, the hybrid song, and it is a mystery to us. The speculation that could have happened is that we know from work that is done in similar birds who learn their song very early in life from in Germany, from their work on captive birds, not Darwin's finches, they've been able to show that if a bird learns one song and has not heard that song when it starts to sing, then it will take information from the song that it learned when it was very young and the song round about it, and actually alter its song in that way. This possibly could have happened with the big birds, but that's pure speculation, we don't know. Yeah.
So the events you talk about are very sudden events with a large magnitude of change, like the big El Nino.
Yeah.
Could you comment on how these different environmental change, these forcing events, what the difference might be between large magnitude changes that maybe happen more gradually to those that happen more suddenly, and what effect these have on adaptation and evolution?
Right.
The question about gradual change, I think, is a really important one in the light of the likelihood that the global temperatures are rising, and there's going to be significant selection pressures on all sorts of organisms, from microbes to every macrobe that you can think of, from plants and animals and so forth. And it's a great challenge to us as evolutionary biologists to try to anticipate what's going to happen in the light of almost certain increasing thermal change. However, having said all of that, and that is an honest statement, from the Galapagos so far we can say almost nothing about that particular kind of environmental change, and the reason is temperature fluctuates all over the place from year to year, and if there's a signal of global warming, a very, very gradual increase, we're not seeing it yet. What we are seeing is changes and no changes. And when I say no changes, no detectable changes. So from year to year, there's virtually no change, typically. And then once in a while, as I mentioned, there are some major perturbations of the environment, and they do result in selection. Is that a message for all of us elsewhere in the rest of the world? Well, I think so, because in addition to gradual climate change that is forecast by the International Panel on Climate Change, there is the forecast of an increase in frequency of extreme events, or maybe even increase in the intensity or the extremeness of extreme events. And so for anticipating what's going to happen in continental regions, or at least elsewhere, let's say outside Galapagos, I think what our study has done is present a model of what could happen. Thank you.
Coming back to these inbred birds, so the pedigree looks like that of the kings of Spain in the 16th and 17th century. So it seems to have been a very unusual event, and you mentioned that there is no inbreeding depression apparently. So you think it's because the populations are small and then there is no inbreeding depression in general, or it's just a chance event, the second chance event for you?
Yeah, so far they haven't shown a Habsburg lip or anything like that, but no, I think actually probably what one of the reasons why they haven't is that they've started off through this introgression hybridization. So they start off with an enormous amount of variation that's going, so they have this when they've been going through. There have been other cases where birds have become very inbred. There's a mockingbird that has been on Champion Island with a population of about 20 pairs at the most, and fluctuated up and down. And they have been doing extremely well for 100 years. I think it's a very interesting problem that it seems that there are some populations of birds that don't show the inbreeding depression that you get in mammals. And I'm just sort of, I think it's an interesting problem. One of the things that could be happening is that birds have, and the Darwin's finches have, 63 chromosomes or something like this?
Well, no, 76.
76 chromosomes. So there's a huge amount of recombination, you know, every time. So this may be keeping you start off with a lot of variation in the genome, and then this also may be maintaining a lot of this recombination, maybe maintaining a lot of genetic variation. I think it's an interesting problem. Yeah. How the difference between the inbreeding depression in mammals and birds.
When the environment is modified, there is a change in the direction of selection on big size, but in view of the role of introgression that you've shown, do you think that there could also be a new selective pressure on mating preferences?
Didn't quite answer.
Like if you need to have a small beak, then you have a new selective pressure on choosing a mate with small beaks?
Yes, it's a selection pressure on beak size.
Yes.
If you have a different beak size, is there a new selection pressure on choice of mates? Do you want to answer that, or shall I answer?
Let's try this one. Hello? No.
No. You start first.
So the question of, with a change in beak shape, should there be altered selection pressures on mate choice? Yes, if there's very, very fine discrimination on the basis of beak size. But so far, the shift that we have observed, dramatic as it looks like in ecological terms, has had no reproductive consequences. And actually we doubt it too. I think if you can imagine, remember the plot that we both showed about population starting off in beak morphology at about this point, and then going up and across and down to over here. That transition from starting to finishing point is nowhere near as big as it would be if it was from one species to another. We'd have to go fairly far out before I think we would see any evidence of a discrimination on the basis of beak size. Certainly species do discriminate on the basis of beak size, but let's say a semi-species, virtually not at all.
I'll just add to that. It really is song in association with beak size. So if they're singing, you know, this is how we get the hybridization. So if if they are singing in association with beak size, then we can, we will, within a population, then we will do this. And within the population, we do get, where we get this enormous variation after this, let's say in the Scandens population, we get large Scandens mating with small Scandens, and so we don't see anything like this.
Yeah, the question is about the role of the very big species, Magnorostris, in it's doing lots of harm to Proctis species, if I understood well, and less to the big birds.
Yes.
But in the morphological space, you see the big birds closer to Magnorostris than the Proctis. So what mechanism could be behind this kind of non-linear niche relationships?
Yes, I mean, we think that they are much closer to Magnorostris. They are different from Magnorostris. I mean, Magnorostris are even bigger than that. And this is where we think if there was any introgression through learning of the wrong song, we could see this with Magnorostris. So far, we haven't. The difference between Magnorostris is a little bit more of a specialist than the big birds. The big birds feeding is, they are extreme generalists. I didn't mention this at all, but they are. They have the same, we've looked at the efficiency with which they eat this large tribulus seed. They actually are just as efficient as Magnorostris in eating the tribulus seeds, and they're smaller, so they may need less during a drought to actually maintain themselves. That's one possibility. The other thing is that they take a puntia, like the Scandens do, and they can also feed on small seeds as well. So they have they're real omnivores. They have a huge range of, so they could do rather well if there was a crunch between Magnorostris and the big birds. But we haven't actually experienced that yet. Yeah.
So the best of both niches, maybe a way of putting it, but they lie between Magnorostris and Fortis. They have an advantage over Fortis in being able to crack open those large hard seeds really quite efficiently. And they have the advantage over Magnorostris in being able to exploit all those cactus foods, somewhat like a cactus finch, not as well as a cactus finch, but somewhat like. So they are intermediate generalists that have an advantage of both over both.
Thanks very much again for this excellent talk. Of the many extraordinary things that you said, there's one that I'm finding particularly extraordinary, which is the idea that a single bird can go into an island and start a lineage. I mean, in my mind, you kind of need a flock of birds, goes into an island, starts a lineage. How did this bird manage to make with 3 different females with having a different song and different appearance?
No, that's our question too. It is extraordinary. I would never ever have thought that a single bird could start this. Part of the reason, well, as I said, it did take a long time to breed, and part of the reason was it bred, I think, later in the season when other birds had already been, and it was, you know, it sort of took the best of a bad situation, or maybe the females were having a tough time getting a mate. And so it could be that they, first of all, it mated with, and it did produce some offspring, but none of those offspring went on to produce birds. But with the Fortis, they did go on to produce offspring which later bred. And the only reason that I can think of that this was late in the season when it first started this, and then once they breed successfully, very often the next year they will breed with the same female. So that's, but it is, as you say, it's not what I expected either. Yeah. Do you have anything to add? This is what happens when you go somewhere. You see things that you don't expect. And sometimes students have said to me, what would you, what would you recommend? And I would always say, follow your exceptions, because the things that you don't expect are the things that most often lead you to new discoveries. So by following what you see something and you think, this is impossible, I never expected this, you follow it, and then it can lead to very exciting discoveries.
You spent 40 years, a long time in the field. I understood that you could read all the birds in the small island. Do you think you can continue, or somebody will continue this study for such a long term, hopefully? And how do you see it in the future with the current movement in science?
Okay, I'll answer this, and then you can. It was the question was carrying on for 40 years, yes, which is very difficult in the current science. Yes, actually, we haven't been back to the islands for a couple of years. We actually stopped at 40 years. We're really getting quite ancient. And it is very difficult. None of the exciting things we have found, we put in a grant application. And so we have gone from, as everybody does, from 2-year grants to a year grant to 3-year grant, 1 5-year grant, that sort of thing. You're hopping through. But usually there's always been a question that it has been interesting enough that we have put in a research proposal. I mean, we haven't said we want to find the origin of a new species or anything like this, but there have been other things like the competition between things that we can answer from a research proposal. So it's not been easy. And I think in the States anyway now, they do have long-term, they do have a, what is it, the
LTER, long-term environmental monitoring.
Yes, which you can get from the National Science Foundation. And they've been, we have not had one, but other people for long-term monitoring really have had this, which can take you over this.
So I would add to that we need luck. You probably do in France as well, a little bit maybe. But anyway, we definitely need luck because there are so many other people competing for relatively small funds. What has worked to our advantage is a rich system with not just a single question that could be addressed, although we pose it in a lecture like this as a series of single questions that we then attack. In fact, we were aware right at the beginning that we had a mixture or variety of topics to consider. There was the topic of evolution, which we couldn't anticipate in a particular year, but we had a very good reason to believe that it was going to happen. We had the question of the competition between species that I've mentioned. How do species keep apart? The answer Rosemary dealt with. And then a question that Rosemary touched on very briefly was the issue of why do some populations vary so much in morphological traits compared with other ones. So we knew we had this richness, but we also knew that we didn't have enough time to answer all the questions in a short amount of time. So it was possible to go from one 3-year period to another, asking different sets of questions, and we hoped that they would be at the forefront of scientific investigation, and fortunately, most of the time they were exactly that. So to summarize all of that, I think one technique that could be used for doing that, what we would do now if we were applying for research, is take a new angle on the topic, a broad topic of evolution, take a new angle, like a molecular genetic angle that we hadn't taken beforehand, and then either collaboratively or by ourselves approach that one in the next funding period of 3 years, drawing upon all of the work that's been done beforehand. So building upon the foundation that's been laid by prior research to ask basically new questions. I know it's easier said than done, but that would be the principle that would guide us.
While the microphone's going up there, I'll just say that our work in collaboration has, especially with the geneticists, has been absolutely amazing. We couldn't have possibly done that on our own.
So, considering the time of divergence of real species and the time it took for them to converge back together, you say it's quite difficult to finally have two clearly separated species.
Can you? Okay, it's considering the time that it took. Is it and
especially in symmetry
especially in sympatry, where you would think that there would be more convergence going on rather than a new species coming in. I think you're right, and I think the way to look at the whole radiation with that in mind, and especially when Peter showed you the work that had been done, which showed during the ice ages how islands had come together and separated, come together, separated. There would be times when species were converging, just as we saw with Fortis and Scandens. But then also with introgression, as we've shown, there could be populations moving into completely new ecological conditions. So both would be going on. And so I think you get this, if you were to look back in time and take snapshots through time, even through the last, let's say, let's say the last 5 glaciations or something like this, then I think you would see examples of convergence and also lineages going off on different trajectories. I think you'd see both.
I would just add that I think there are many, in this particular system, many cases of failed divergence. The incipient divergence in Allopatric, leading to the sympatric condition of partially differentiated populations. They're not different enough to maintain themselves as separate entities. Interbreeding would have occurred fairly frequently, and a fusion back into a single state would have been the outcome. What Rosemary described with Fortis and Scandens is rather different. These two species really were reproductively isolated for a long time before, with the triggering of an environmental change, they started to fuse together. And it gives us an insight into what must have happened, we think, many times, naturally in the radiation. A few species formed, and many attempted speciation events collapsed, we think.
So I've seen that most of your work was done on beak-related traits. And I was wondering if this focus was due to the fact that you think this was the strongest selection signal you could get on any specific trait, or just because it's got a clear causal process behind selection on it? I mean, it's easy to relate that to variations in seed size due to environmental change.
Yes, I mean, beaks are the tools that we have hands. We have hands, but for a bird, the beak is absolutely vital. It's a tool that it uses to pick up food. So that's why we chose beaks. We also chose beaks because they were measurable. But there are other things, and especially in other organisms, in other situations that might stand out more. So, for example, physiological things. Probably particularly in mammals, it may be more important to look for physiological traits, or, you know, so, but beaks are the very obvious thing in birds to pick. So that's why we chose beaks. We also measured other things, but it was the beaks that were actually moving in these selection events. Yeah. Mm-hmm.